Hi everyone, my name is Matt, and to tell you about my research, I'm going to begin by telling you a little bit about cells. All living things are made up of cells. Cell division, which is the process by which one parent cell splits to create two daughter cells, is at the heart of the growth and reproduction of living organisms. These cells that make up living things are themselves made up of molecules that you've likely heard of: DNA, sugars, fats, and proteins. These molecules control the identity and the behavior of cells, so when cells divide, they have to ensure that their daughters inherit the appropriate set of molecules. Sometimes this means that the daughters inherit the same molecules in a symmetric division, and sometimes this means that the daughters inherit different molecules in an asymmetric division. On the slide here, I've diagrammed a symmetric cell division. Signaling proteins are one important class of biological molecules and the focus of my research. As drawn on the slide, signaling proteins are embedded in the membranes of cells, connecting the inside to the outside and allowing cells to communicate with their neighbors and with their surroundings. It is this communication that coordinates the efforts of the trillions upon trillions of cells that make up your body. Like the other molecules that make up cells, signaling proteins have to be appropriately inherited during cell division. However, we know very little about what controls the movement of signaling proteins as cells divide, and therefore very little about what regulates their inheritance by daughter cells. My research has revealed a link between the regulators of cell division and the movement of signaling proteins during cell division. I found that CDK1, a well-known protein responsible for initiating cell division, protects signaling proteins from being degraded as cells divide. Because of CDK1's activity, signaling proteins are stored inside the cell during cell division, as diagrammed on the top right. We think that this storage is important because it allows signaling proteins to persist through cell division and be appropriately inherited by daughter cells. This relationship between the regulators of cell division and the movement of signaling proteins during cell division provides a robust and elegant mechanism to explain the regulation of the inheritance of signaling proteins. Because CDK1 always coincides with cell division, dividing cells will automatically activate this CDK1-controlled pathway to store signaling proteins. This type of research is important because both errors in cell signaling and errors in cell division are hallmarks of human cancers. My results help us to better understand the relationship between signaling and cell division in healthy cells, which in turn informs our understanding of how these processes can fail in cancerous ones. Thank you.